This lecture, what we're going to be discussing is synaptic transmission. So in the previous exam material, what we covered was the brief history and the brief introduction of what neuroscience is and does, the different types of cells that exist within the central nervous system, particularly in the brain. In chapter 3 and 4, we talked about the neuronal membrane resting potential as well as the action potential. And so in this section of the course, what we're going to be talking about is introducing what happens when the action potential gets down to the axon terminal. So this action potential travels down towards the terminal, then what happens? That's going to be the subject of this lecture, which is all about synaptic transmission. So synaptic transmission is the area of the neuron where there is information transfer occurring. So you have an action potential which travels down the axon, which is this large projection neurite, and goes down to the axon terminal, which is the end of the presynaptic, or the neuron that comes before the synapse. And remember that the synapse is a small gap in between your presynaptic and your postsynaptic cell. So synaptic transmission plays roles in all the operations of the nervous system, and when we talk about the interaction of drugs in the brain, drugs are having all of their effects at the level of the synapse. In 1897, Charles Sherrington coined the term synapse. And one thing that we're thinking about when we go into the idea of synapses and having these gaps in between these cells is in 1921, Otto Louis did this really cool experiment with frog hearts. And in this experiment, he was able to show that there is chemical neurotransmission occurring. It's not just all electrical. And in the late 1950s, Firshpan and Potter showed the presence of electrical synapses. So there are two different classifications of synapses within the brain. You have chemical synapses, which are going to involve the release of chemicals from a presynaptic to a postsynaptic cell. And then you have electrical synapses, where you have a tight junction occurring between two cells. So in 1921, Otto Louis did a really cool experiment. So the hypothesis was that neurons release chemical substances that can influence the activity of the heart, and in the same regard can influence any kind of tissue that has a respective receptor. So what Otto Louis did was he dissected hearts from two different frogs and placed them in chambers filled with saline solution. So here's the chamber, here's the saline solution, here's heart number 1. Note, heart number 1 has the vagus nerve attached, so it's intact right here. And frog heart 2 is in the chamber with the vagus nerve removed. So at the conceptual level, it was already known at this time that the vagus nerve has an inhibitory effect on heart activity, meaning that it slows down the heart. So what Otto Louis did was he electrically stimulated the vagus nerve of heart 1. By electrically stimulating the vagus nerve, you're activating the vagus nerve, causing heart 1 to slow down. So here, this is demonstrated down here. So stimulation of vagus nerve of heart 1, here's the response that shows to be diminished once the vagus nerve is activated. Now, you do not stimulate any nerves that are attached to heart 2. Remember that there's no nerve attached here. So what Otto Louis did was he stimulated the vagus nerve of heart 1 in heart 1's saline container. So what was done was the strength and number of beats in heart 1 was recorded before and after electrical stimulation. That way we're able to confirm the fact that, okay, stimulation of the vagus nerve led to a decrease in heart response. So next, he removed a sample of the saline solution that was found in and around heart 1 and then transferred that to heart number 2. So what he did was he took a sample of the saline solution that was in and around the heart and simply moved it and placed it into saline solution 2. Remember that in this area, we don't have the vagus nerve attached. And then what happened was he recorded the response of heart number 2. So at this point, we added saline from chamber 1. So all we did was add saline. What was found was that you see there's a change in heart activity, a decrease in heart 2's response. Now, this is great evidence, and this is saying that, okay, there are chemical transmitters, there are chemicals at work that led to a decrease in heart contractions. That has to be the case because, again, we didn't electrically stimulate this heart at all, right? We didn't stimulate heart number 2 because the vagus nerve was removed. So what was causing the decrease in heart 2's response? Well, it has to be something inside the solution of heart 1. So if stimulation of the vagus nerve resulted in the release of some chemicals onto this first heart, then those same chemicals, which would probably be diffused within the solution, should have an identical effect in heart number 2, and that's exactly what was seen. So the direction of information flow will generally go in the form or in the flow of one direction, from a neuron to a target cell. So the first neuron is known as the presynaptic neuron, and the target cell is known as the postsynaptic neuron. So here, if we have an axon terminal connecting to a dendrite, let's say, this space is known as the synapse. The cell that comes before the synapse is known as the presynaptic cell. And the cell that is after the synapse is known as the postsynaptic cell. So here's the general anatomy of a synapse. So you have the axon terminal right here that contains synaptic vesicles filled with neurotransmitter, which are the chemicals that will aid in communication. The process of exocytosis is going to lead to the excretion of these neurotransmitters into the synaptic cleft, which is this empty space right here. For the most part, we think of the synapse as empty space, but there are different little fibers that are able to essentially keep this formed synapse in place. So basal lamina are fibrous extracellular proteins which help to essentially bind these membranes together. So on the postsynaptic cell, which is again the cell that lies after the synapse, you have these proteins, these receptors, that are going to aid in neurotransmitter recognition. Then those are going to open up ion channels such as potassium, sodium chloride, and have an effect on the membrane potential of the cell. See, I hope that you begin to understand and realize that everything here is connected, and understanding Exam 1's material is going to be very beneficial in going into this chapter, because assuming you have done well, and assuming that you have a good understanding of all the material from chapter 1 through 4, that's going to help us build on that by looking at neurotransmitter systems and synaptic transmission in depth. Now, chemical transmission and chemical synapses are the predominant species in the nervous system, but we will spend a little bit of time talking about the presence of electrical synapses because they do exist. Now, electrical synapses allow direct transfer of ionic currents from one cell to the next. Now, these occur in numerous cell types, including glia, smooth and cardiac muscle cells, as well as epithelial cells, and in neurons. So, in electrical synapses, you have the dendrite of one cell connecting to a gap junction of a particular area of another cell. Now, what happens in this area is that you have a direct communication between the contents of one cell and the next through processes known as gap junction channels. So if you have an axial potential changing the membrane potential of one cell, that will lead to the direct transfer of ionic current into another cell. You're not having to go through a chemical transmission before you get into the second cell. So the gap junction is made up of channels. You have a connexon, which is formed by 6 connexins. And then 2 connexons combine to form what we know as the gap junction channel. So you have 2 connexons which form this channel between the 2 cells. And then each of those connexons is formed by 6 connexins. So in the form of gap junctions in electrical synapses, the cells are said to be electrically coupled. The flow of ions from one cytoplasm to another cytoplasm. So the pore has a relatively large diameter, which allows for the passage of ions and many small organic molecules to go between these cells. So here's our anatomy of a gap junction, where we have these gap junction channels which exist, which are made up of 6 connexins, which forms 1 connexon, and then 2 connexons forms the entire gap junction. So here's the connexin, 1, 2, 3, 4, 5, and 6. 6 connexins forms up 1 connexon, and then 2 connexons are going to form your whole gap junction channel. So this is going to allow for the direct transfer of ions and small organic molecules between a presynaptic and a postsynaptic cell. So unlike most chemical synapses, electrical synapses show bidirectionality, meaning that electrical current can pass through these channels both ways. So we call them, again, electrically coupled. Now, electrical synapses allow for very fast transmission. So postsynaptic potentials in the receiving neuron are pretty quick. So if a second neuron produces an action potential, remember, due to bidirectionality, a postsynaptic potential can occur as well in the first neuron. And we'll talk about the idea of synaptic integration in a lot more detail in the next chapter and at the end of this chapter. But several postsynaptic potentials can occur simultaneously to excite a neuron, thus causing an action potential. And again, remember from our last chapter, in order to have an action potential, all you need to do is get to negative 55 millivolts. So this is definitely noted in electrical synapses where neurons can make electrical synapses with numerous other neurons. So here in an electron microscope diagram, we see this small connection known as a gap junction. So electrical synapses are very common in early development. So during pre- and postnatal brain development, where these gap junctions allow for neighboring cells to share both electrical and chemical signals. And the goal of this and the overall reason for this is to help coordination of growth and maturation. So a synchronization of oscillations and action potentials, which allows for coordination in responses. So the roles for these gap junctions vary from region to region. And one area that they're definitely found in is where normal function requires a high level of synchronization. So particularly in the inferior olive in the brainstem, where axons are sent to the cerebellum, this is important in motor control. And this helps to coordinate and synchronize activity of these neurons, helping to control the fine timing of motor control. Now, most synaptic transmission in the mature human nervous system is going to be mediated by chemical synapses. So you have secretory granules, which are larger vesicles containing soluble proteins, and you have large dense core vesicles which contain neuropeptides. So here's the general anatomy of a synaptic neurotransmission site. You have your soma, your dendrite of your presynaptic. So this cell over here is your presynaptic cell, making a connection to a postsynaptic cell. So you have the axon terminal, which is the end part of the axon from the presynaptic cell. The active zone and the postsynaptic density are the sites of neurotransmitter release and where neurotransmitter receptors are respectively. Synaptic vesicles are what store neurotransmitters, and the synaptic cleft is this brief empty space. But remember, it's not entirely empty. So chemical synapses have a large amount of mitochondria due to the high energy demand. You have vesicles, which are going to store the neurotransmitters, and dense core vesicles, which help to store neuropeptides. Remember from earlier chapters how we get the peptides down through anterograde transport down from the soma, where transcription and translation occurs, down to the terminal itself. Here you have neurotransmitter that is then released into the cleft, which can then bind to proteins known as receptors. So the types of synapses all depend on which part of the neuron is postsynaptic to the axon terminal. So the general rule is, in naming these, you can have a presynaptic element, whether that be an axon or a dendrite, making a connection to a postsynaptic element, whether that be a dendrite, a soma, or an axon, and then add an ick to it. So most often, a central nervous system synapse involves an axon, remember our axon terminal, making a connection to a dendrite. That's an example of an axodendritic connection. An axon to a dendrite, axodendritic. So the first part of that word is where the presynaptic element lies. The second part of the word is where it's making a connection to. But other types of connections exist, including axosomatic and axoaxonic, where the axon is making a connection directly to the soma, and where an axon is making a connection to an axon. An axospinous synapse is an axon to a dendritic spine. And then even dendrodendritic synapses exist, where dendrites are able to make connections with other dendrites. So here are brief examples. So an axodendritic synapse is where an axon is making a connection to a dendrite. This is where the synapse lies. Axosomatic is where you have an axon making a connection to the soma. And an axoaxonic is where you have an axon terminal making a connection to another axon. Now note, presynaptic terminals can be recognized by their mini vesicles, and postsynaptic elements have postsynaptic densities. So note here, these are different examples of our presynaptic terminals and how they can look. You don't just have a general shape like this. You can have an axospinous connection where you have an axon making a connection to a dendritic spine. You can have an axon that splits here with numerous active zones. You can have several active zones, particularly around an axon and postsynaptic elements. And then you can also have presynaptic terminals making connections to several different areas. So A represents our axospinous. B is our axosomatic, where we have an axon making a connection directly to the soma of the receiving neuron. In figure C, we have a large axon terminal surrounding a postsynaptic soma. Remember that the cells are in 3D, right? So you can have this large axon surrounding this soma, and then there be dendrites coming up here in 3D space, right? So larger synapses will have more active zones. So there are two different categories for synaptic membrane differentiations. You have Gray's type 1 synapses, which tend to be asymmetrical, and Gray's type 2 synapses, which are symmetrical. So Gray's type 1 synapses, where the postsynaptic side is thicker than that on the presynaptic side, these will usually be excitatory synapses. And in Gray's type 2, which is symmetrical, where you have symmetrical membrane differentiations, which are similar in thickness, these are usually inhibitory. For the most part, I'm not going to use this terminology all too much, but these are different ways that synaptic membrane differentiations can be categorized in the central nervous system. Now, synaptic junctions do not just happen in the brain. They're found in a lot of other areas of the body, including glands, smooth muscles, and even the heart, where you have innervation by axons that lie in the autonomic nervous system. So chemical synapses between axons of motor neurons of the spinal cord and skeletal muscles occur at what's known as the neuromuscular junction. So that's a combination of the nervous system and the musculature system. Now, the neuromuscular synaptic transmission is very fast and very reliable, where an action potential in the motor axon will always cause an action potential in the muscle cell that it innervates. And this is one of the largest synapses in the body with a large number of active zones. Now, the postsynaptic membrane, which is known as the motor end plate, contains a series of shallow folds that are precisely aligned with the presynaptic active zones. This is why an action potential in the motor neuron will always cause an action potential in the muscle cell it innervates. The postsynaptic membrane of those folds are packed with receptors that are going to be receiving information from acetylcholine that is released. This is the neurotransmitter that is released here. So acetylcholine, the neurotransmitter, is focally released onto a large surface of chemically sensitive membranes that are going to be receptive for receiving that neurotransmitter. And the studies of the neuromuscular junction have helped to establish the principles of synaptic neurotransmission with numerous implications: diseases, drugs, poisons, chemical warfare, etc. So the neuromuscular junction, or the NMJ, acetylcholine is the major neurotransmitter released here. And this is going to be very important. So at the neuromuscular junction, acetylcholine is the neurotransmitter that is released. So here's a motor neuron, which is an efferent neuron, which is sending information out into the periphery. Here's your axon terminal, making a connection to the motor end plate, which is right underneath it at the postsynaptic region. You have your synaptic vesicles that are going to be filled with acetylcholine. Here's your cleft, and then receptors, which are found directly underneath the presynaptic terminal. This is why it's so reliably known that when acetylcholine is released, there's always going to be action potentials at the muscle fiber. So nerve gases that act by blocking acetylcholine esterase, the enzyme that breaks down acetylcholine, will cause continuous contraction of muscles, and it can also affect glands and organs. Acetylcholinesterase, abbreviated ACHE, is a very important enzyme because you don't want acetylcholine continually stimulating and contracting muscle tissue, right? You want that acetylcholine to be regulated and broken down and removed so that continual muscle contraction isn't a worry. However, if your diaphragm muscles and your peripheral muscles are consistently in a state of contraction, you're going to be unable to function, and ultimately you'll die. So certain nerve gases will act to inhibit acetylcholinesterase, and they're known as acetylcholinesterase inhibitors, meaning that they're preventing acetylcholine from actually breaking down. So a brief overview of chemical synaptic transmission. These are going to be steps that you're definitely going to need to understand and know, because this is how everything works in the brain. So you have synthesis of neurotransmitters, which we're going to get into a lot more detail about in the next lecture, where we talk about all the neurotransmitter systems. But the basic key ideas here that I need you to understand are as follows. You have the transmitter of any given type, so GABA, glutamate, dopamine, serotonin, norepinephrine, acetylcholine, etc. You need that neurotransmitter to actually be created, right? And you need it to be stored in vesicles. So we need to create neurotransmitters and load those neurotransmitters into synaptic vesicles. Now step 2, in action potential, which we are all experts on at this point, will invade the presynaptic terminal. So an action potential will travel down to this terminal. When the action potential comes here, it will lead to voltage-gated calcium channels opening up. Now, what that voltage-gated calcium channels opening does is it leads to an influx of calcium through those channels, which then will cause these vesicles to fuse with the presynaptic membrane in a sort of zipper-like fashion, causing the neurotransmitters to spill out into the synaptic cleft. Now, once those transmitters are released in the process known as exocytosis, those transmitters can do a few things. So the transmitters can bind to receptor molecules that lie in the postsynaptic membrane. They can lead to the opening or closing of postsynaptic channels. This will then cause excitatory or inhibitory postsynaptic potentials that can lead to change in membrane polarization of the postsynaptic cell. And the last part is where vesicular membranes from the plasma membranes are retrieved and recycled, where additional neurotransmitter can be placed in. Now, another big idea here is that you can't just have these neurotransmitters here forever, right? You need some mechanism whereby those neurotransmitters are going to be removed from that cleft, where they're not continually going to activate these receptors. And you have a few different ways that these neurotransmitters can be removed. You have diffusion, enzymatic degradation, or transport and reuptake. So in general, brief neurotransmitter categories include amino acids. So these are small organic molecules which will be housed within inside vesicles. So examples of these include glutamate, glycine, and GABA. So different neurotransmitters are released under different conditions. And that's going to be a big key idea, especially when we start talking about the different neurotransmitter classes. You have amines, which are small organic molecules which are going to be found within inside vesicles. Examples of these include dopamine, acetylcholine, histamine. And then peptides, which are short branches and short chains of amino acids. So these are small proteins. These are housed within secretory granules. So examples of this include substance P, cholecystokinin, dynorphin, enkephalins, etc. So here in table 5.1 are just some brief examples of major neurotransmitters that fall within these general categories of amino acids, amines, and peptides. For this class, you're not going to be required to memorize all these examples, but you should be familiar with these major categories. So different neurons release different neurotransmitters. You're not going to have one neuron in the nervous system releasing both acetylcholine and dopamine at the same time. You have dopaminergic neurons, which are going to release dopamine. You have acetylcholinergic or cholinergic neurons, which release acetylcholine. So fast forms of synaptic transmission are mediated mostly by glutamate, GABA, or glycine. Now, this transmission lasts from about 10 to 100 milliseconds, and here are these classes of amino acids. glutamate, GABA, and glycine. Acetylcholine mediates fast synaptic transmission at all of the neuromuscular junctions. So here's acetylcholine. And slower forms of synaptic transmission can utilize neurotransmitters from all three of the chemical categories that we talked about, the peptides, the amines, and the amino acids. And these can last from hundreds of milliseconds to minutes. And so peptides are really good at slower forms. And when we start talking about also endocrine transmission, particularly with hormonal transmission of information going through the bloodstream, this is definitely a slower form of transmission that occurs within the nervous system. So neurotransmitters are synthesized and stored in synaptic vesicles. So amino acid neurotransmitters are abundant in all cells of the body because amino acids are abundant in our cells. So other neurotransmitters such as GABA and the amines are made primarily in the neurons that release them. So that makes sense. If you're a GABAergic neuron, you're going to have enzymes that are going to synthesize and create GABA, right? So specific synthesizing enzymes are found in those neurons that synthesize those neurotransmitters. So that's a good clue as to the type of neuron that you're in. If you're looking at neurons and you find that you have synthesizing enzymes that are important for acetylcholine, well, you're probably in an acetylcholinergic neuron. Now remember from our discussion on transcription and translation, peptide synthesis and transport, because we're looking at proteins, are going to occur in the soma. So transcription in the nucleus, the mRNA transcript that leaves the nuclear envelope, then we have translation that occurs outside of the nucleus in the ribosomes. Now these peptides will be synthesized and transported down in an anterograde fashion in secretory granules that will then be housed here in the synaptic terminal or in the presynaptic terminal. So the general path that's taken here is we have precursor molecules, which will then undergo a synthesizing enzyme reaction to be a neurotransmitter molecule. Then transporter proteins will allow for those neurotransmitters to be placed inside a synaptic vesicle. And the most abundant neurotransmitters are going to be synthesized directly here in the presynaptic terminal. So that's a big idea to understand, is that small neurotransmitters, like most of the amino acids and the amines, are going to be synthesized directly within the presynaptic terminal. Neuropeptides are going to be synthesized in the soma. Now, how do neurotransmitters, how do they communicate? How are they released from these vesicles? So we know that they are placed in vesicles in the presynaptic terminal, but how do they get expelled? This is through the process of snare proteins. So neurotransmitter release is triggered by an action potential that comes into the presynaptic terminal. So we talked about that. This whole process is known as exocytosis. When that action potential comes down into the presynaptic terminal, voltage-gated calcium channels open in the active zones. This leads to a large inward driving force on calcium. Calcium is highly concentrated on the outside in the extracellular space, so when these voltage-gated calcium channels open, you have calcium flow in, and calcium is Ca2+. This is the process known as exocytosis, which is stimulated by the rise in the intracellular calcium levels. Now, proteins alter conformation when activated, so we have a fusion of vesicular membrane into presynaptic membrane that occurs at the active zone, leading to neurotransmitters then being spilled out. Now, this is a very quick process because calcium enters that active zone precisely where vesicles are ready and waiting, directly for them to be released. Now, vesicle membrane can be recovered through the process of endocytosis, and this recycled vesicle is going to be refilled again with neurotransmitter through vesicular transporters. And these vesicular transporters, the job of these vesicular transporters are to take those neurotransmitters that are synthesized here in the terminal and place them into the vesicles. Now, peptides in secretory granules are also found in the presynaptic terminal, although they're created and synthesized in the soma. Release of these peptide markers are generally requiring high-frequency trains of action potentials, so that the calcium inside the cell throughout the terminal can build to the level required to actually trigger the release. So, the snare family of proteins are vital for specific binding and fusion of membranes. And the process of exocytosis is mediated by the formation of what's known as the snare complex. So here we have our vesicular membrane that has different snare proteins: synaptotagmin, synaptobrevin, SNAP25, and syntaxin. There are things known as T snares, which are syntaxin and SNAP25, and then you have V snares, or vesicle-associated snare proteins, so synaptotagmin and synaptobrevin. Synaptobrevin on the vesicle will interact with syntaxin and SNAP25 on the target membrane. Now what happens in this interaction is that you have essentially a tying together of these proteins where you allow for fusion of the vesicular membrane. So here we have the membrane of the vesicle, and allow this to fuse with the membrane of the plasma membrane. And so briefly, I'll show you just this video really quick right here, which demonstrates the process of exocytosis. Here we have synaptobrevin, syntaxin, and SNAP25. And here we have a vesicle that contains neurotransmitters. So synaptobrevin, SNAP25, and syntaxin are vital. And the whole complex of the snare proteins is vital for neurotransmitters to actually be released in the first place. Now, we're focusing here on synaptobrevin as a vesicular snare protein and SNAP25 and syntaxin as target snare proteins, but what about synaptotagmin? Well, synaptotagmin have been identified as major calcium sensors that trigger the fusion to occur. So the cytoplasmic region is composed of two different domains which can bind a total of 5 calcium ions. Now the binding of those calcium ions is thought to act as a switch, allowing for the interaction of these two domains with phospholipids to occur. And remember, these vesicular membranes and plasma membranes contain phospholipids. So calcium binding to synaptotagmin, remember, when the action potential comes down to the terminal, that leads to voltage-gated calcium channels opening, and calcium is able to flow in. Binding of calcium to synaptotagmin will trigger the release by stimulating synaptotagmin binding to a core fusion machinery, which then will mediate the fusion of these proteins. Now this is a very intricate system. By no stretch of the imagination do I want you to be experts at identifying all these different proteins, but you should be very familiar with the idea that calcium is important. So snare proteins are also substrates for botulinum and tetanus toxins, where under the case of botulinum toxin, these snare proteins are actually cleaved. And when you cleave these snare proteins, you are preventing them from actually functioning correctly. So bacterial proteases that are potent inhibitors of neurotransmitter release will prevent neurotransmitters from actually being released into the synaptic cleft. So in a normal neurotransmitter release system, you have synaptobrevin on the vesicular membrane and you have SNAP25 and syntaxin on the target membrane. These will fuse, allowing for neurotransmitters to be released. Now, as is the case in Botox, or in the case of botulinum toxin, this chain will actually cleave snare proteins. And if you cleave them and you actually break them apart, you actually lead to the fact that the membranes do not fuse together. The snare complex doesn't form, therefore the vesicle cannot form and fuse with the presynaptic membrane, thus preventing any kind of neurotransmitter from being released. Now, what happens when neurotransmitter is released? So we have neurotransmitter that lies inside vesicles here in the presynaptic terminal. And then we've already said that neurotransmitter can then spill out into the cleft. So here we have all this neurotransmitter. What happens to it? Well, it binds to proteins known as receptors. And these receptors can be classified into two different types: transmitter-gated ion channels, also known as ligand-gated channels, or G-protein coupled receptors. So we'll start off with ligand-gated channels. So ligand-gated, and a ligand is just something that can bind to something else. And this is also, again, known as transmitter-gated ion channels because in order for the channel to open, it needs to be bound to a neurotransmitter. So ligand-gated channels are membrane spanning, and that makes sense because in order for ions to go from the inside to the outside or vice versa, you need a channel that goes across the membrane. Here's the outside of the cell, and here's the inside cytoplasm. You have 4 to 5 subunits that come together to form a pore. And so we talked briefly about this in chapters 3 and 4 when we were talking about ion channels, allowing for sodium and potassium to flow through. Now, the general mechanism of this is the neurotransmitter that is released from the presynaptic terminal will bind to a binding site here that's present on the protein. This will lead to a conformational change, allowing for the pore to open. Now, this isn't as selective for particular ions as are the voltage-gated sodium and potassium channels. Now, opening of these channels and bringing in ions or allowing ions to exit will lead to 2 different responses: an excitatory postsynaptic potential, which is abbreviated EPSP, or an inhibitory postsynaptic potential, abbreviated IPSP. EPSPs and IPSPs are both transient postsynaptic membrane potentials, meaning that they're short-lived. And in an excitatory postsynaptic potential, we have a depolarization caused by the presynaptic release of neurotransmitter. Examples of this include acetylcholine and glutamate, where we have sodium coming into the postsynaptic cell. And an inhibitory postsynaptic potential is a transient hyperpolarization caused by release of GABA, allowing for chloride ions to come into the postsynaptic cell. Remember, you have about negative 70 millivolts, so if you're allowing for more chloride ions, which carry a negative charge, to come in, you're increasing the amount of negative charge. However, if you're opening up a sodium channel, allowing for more positives to come in, you're creating a more positive or less negative membrane potential. So something like this, here's negative 70 millivolts, here's negative 55. An EPSP would be something like this. Where it's transient and it will go back down to resting state. However, it's a slight depolarization. So if we do a transient hyperpolarization known as an IPSP, would be something like this. Taking you further away from activation threshold. So an EPSP would be generated through neurotransmitter binding, allowing for sodium to flow in. Again, recording the membrane potential in the postsynaptic membrane. So here we see a depolarization, but not enough to actually hit the activation threshold. And an IPSP allows for chloride ions to flow into the cytosol. Remember, you're adding more and more negative ions, so you're making the membrane potential more negative, taking it further away from activation threshold. Now, G-protein coupled receptors, in comparison to ligand-gated or neurotransmitter-gated receptors, are slower, longer lasting, and much more diverse. They have a lot of different actions that can take place at the postsynaptic membrane than ionotropic receptor signaling. Now, here's this term ionotropic. Ionotropic receptor signaling is also known as ligand-gated or neurotransmitter-gated. Metabotropic is what's also known as G-protein coupled receptor signaling. So neurotransmitter molecules will bind to receptor proteins embedded in the postsynaptic membrane. This is the same thing that we've seen already. However, in the case of G proteins, the receptor protein will activate what's known as a G protein. Now, these G proteins are found in the cytoplasm and are found here in the postsynaptic cell, and they're free to move along the intracellular face of the membrane itself. Now, when these G proteins become activated, they will actually activate different proteins. They'll activate effector proteins. Now, these can be G protein gated ion channels, such as, let's say, a potassium channel, or they can be enzymes, which can then synthesize second messengers that can diffuse away in the cytosol. Now, these are known as metabotropic receptors because they can trigger widespread metabolic effects. So, this is in comparison to an ionotropic receptor, where we had an ionotropic receptor here, a neurotransmitter bound to it, allowed for sodium or chloride to flow in, and that's it. You just have a change right here at this given location on the membrane. However, in G protein receptors, you have a neurotransmitter binding, which leads to the activation of a G protein, which can then go and activate ion channels or enzymes to cause changes further down in this postsynaptic cell. Now, one thing to be aware of is that the same neurotransmitter can have different effects depending on what receptor it ultimately binds to. And that's one of the big ideas, particularly in drug chemistry and the interactions of different drugs inside the brain, is that one neurotransmitter can either have inhibitory or excitatory effects, but also, if it is binding to a metabotropic or ionotropic receptor, it's going to have different types of effects, just like if your neurotransmitter is excitatory or inhibitory. So autoreceptors are found in the presynaptic axon terminal. So receptors are not only found in the postsynaptic membrane, they can also be found on the presynaptic axon terminal. Now, these presynaptic receptors are sensitive to the neurotransmitter that is released by those presynaptic terminals, and these are what are known as autoreceptors. So typically these autoreceptors act as G protein coupled receptors that can stimulate second messenger formation. Now, the consequences of activating those autoreceptors can vary from neuron to neuron, and depending on where you are inside the brain. Now, a common effect is inhibition of neurotransmitter release, and in some case, neurotransmitter synthesis. So let's say you have an overabundance of release of neurotransmitter here, and your autoreceptor gets activated. That autoreceptor will say, okay, we have enough of this neurotransmitter being released, we don't need to release anymore. So that might inhibit neurotransmitter release and neurotransmitter synthesis from still occurring. So what autoreceptors do is that they function as sort of a safety valve. So when neurotransmitter around the terminal gets too high, we don't need to keep releasing more and more. So it's sort of a safety valve to be able to say, okay, we've released enough neurotransmitter, we don't need to release anymore. Now, once that neurotransmitter is in the cleft, how do we either recover it or how do we degrade it? We don't want this neurotransmitter to be continually present in the synapse. And we saw how nerve gas, which inhibits acetylcholinesterase, can be really deadly and really bad, right? Because if you are having continual stimulation of those receptors by those neurotransmitters, then you're having too much excitation or too much inhibition, and that's really bad. So you can have the transmitter being diffused away. So it can simply just diffuse away, either be brought back up by astrocytes or diffuse outside of the cleft. That's sort of a non-controlled, I guess, sort of way. You have reuptake, where your neurotransmitter will re-enter the presynaptic axon terminal where it was released from. Now this occurs through what are known as transporters in the presynaptic membrane. Now these can then be reloaded into vesicles or enzymatically degraded. Enzymatic destruction can occur inside the terminal cytosol or within the synaptic cleft itself. And one good example that we've already looked at is acetylcholinesterase. Acetylcholinesterase will break down acetylcholine in the cleft. And then desensitization is where we can actually render transmitter-gated channels due to continual acetylcholine presence actually cause a desensitization or an inactivation of these channels. So acetylcholinesterase will cleave acetylcholine, rendering it inactive. So despite the continual acetylcholine presence, the transmitter-gated channels remain closed because they act in a desensitized manner to this continual presence. So nerve gas results in the acetylcholine receptors essentially becoming desensitized, and neuromuscular transmission ultimately fails. So nearly every drug with psychological effects acts on the synapse, either through stimulating or blocking receptors. So receptor antagonists are inhibitors of neurotransmitter receptors. They block the activity of that given neurotransmitter. Receptor antagonists bind to the receptors and block the normal action of that transmitter. So an example in the acetylcholine family is Curare, which binds to acetylcholine receptors, particularly on skeletal muscle cells, and blocks the actions of acetylcholine, thereby preventing acetylcholine from binding and causing muscle contraction. So an antagonist is pretty bad. it will block receptors. So if you think about like a book and the protagonist or the antagonist is often the villain, right? Receptor agonists mimic the actions of naturally occurring neurotransmitters. So an agonist at the acetylcholine receptor is known as nicotine. This binds to and activates the acetylcholine receptor in skeletal muscle. And nicotinic acetylcholine receptors in the central nervous system, not out in the periphery, can lead to the addictive effects of tobacco use. Now, defective neurotransmission, whether that be overstimulation of neurotransmitter release, prevention of binding, overbinding, etc., can be the root cause of neurological and psychiatric disorders. Now, synaptic integration is the process by which multiple synaptic potentials can combine with one another in one postsynaptic neuron. So most central nervous system neurons receive thousands of synaptic inputs. So when you read your book or you look at different things like PowerPoints showing one or two connections on a neuron, that's vastly oversimplified. You have hundreds and thousands of different inputs going onto a neuron at the same time and at any given moment. And these can act on both ligand-gated and G-protein coupled receptors. Basically, the neural computation that occurs is going to lead to firing an action potential or not firing an action potential. So you have a combination of EPSPs and IPSPs that will ultimately sum together in the axon hillock. If this reaches negative 55, say it with me, you'll fire an action potential. If it does not, your cell will not. So remember, each postsynaptic membrane of a synapse may have from a few tens to several thousands of ligand-gated channels. And how much neurotransmitter is released will affect how many channels are essentially going to be activated. If you have a lot more sodium channels activated, by definition of more and more neurotransmitter release of an excitatory neurotransmitter, let's say, you're probably going to be more likely to fire an action potential, compared to if you have a lot of channels that are being activated, allowing for chloride to flow in. So EPSP summation is the simplest form of synaptic integration. This allows for neurons to perform sophisticated computations. And this integration is where EPSPs are added together to produce a significant postsynaptic depolarization. So spatial summation and temporal summation are two excellent examples of synaptic integration. Spatial summation is where we take EPSPs that are generated simultaneously at different sites, and those can be integrated and accumulate into an action potential. And temporal summation is where these EPSPs are generated at the same synapse in rapid succession. So in the case of an action potential traveling down the presynaptic axon, this will lead to the release of neurotransmitter, which can then lead to an EPSP in the postsynaptic cell. We record the membrane potential down here in the soma. Remember that right here in the axon hillock is where this action potential will actually be triggered. So this is where we need to see if there's negative 55 millivolts, right? Spatial summation is where we have action potentials from other axons coming together at the same time through different locations. So note, different spaces on the postsynaptic neuron. This is spatial summation. It's a summation across space. And here we record the membrane potential. So let's say this might have been, let's say one of these alone might have given us a little small EPSP. However, if we look at them added together, we might have one EPSP, two EPSPs, and then the third one reaches negative 55 millivolts and fires. Temporal summation is the summation of EPSPs across time. So in rapid succession, you have action potential coming right one after each other that will build on each other, crossing threshold, leading to an action potential to occur. Now, this is, again, vastly oversimplified because you don't just have three action potentials occurring, in rapid succession. You might have hundreds, right? Depending on the type of stimulus that is initiating this action potential. So many dendrites have voltage-gated sodium, calcium, and potassium channels. And what these do is that they can act as amplifiers of postsynaptic potentials, which can help to add the current to boost the signal along toward the soma. Now what happens, and I'll go back here, is when you have a signal that comes in at this point, you actually have dendritic decay because this signal essentially has to travel all the way to the soma and get down to the level of the axon hillock, which is going to be that action potential triggering zone. If this area right here reaches negative 55 millivolts, then you'll fire an action potential. However, if you have a signal that comes all the way, let's say this dendrite goes all the way up here, by the time that this gets all the way down to here, you're not going to be nearly the same strength as you were up here. So there are rarely enough ion channels present in dendrites to generate fully propagating action potentials as axons do. Remember, in axons, we're able to regenerate the action potential due to saltatory conduction in the nodes of Ron-Va, which have a high concentration of sodium channels, voltage-gated sodium channels. Now, dendritic sodium channels in some cells can carry electrical signals in the opposite direction from the soma outward along dendrites. And this has implications in learning. And you will learn more about this in a lot more detail in Psych 376 and in other courses that focus more so on the neurochemical basis of learning and memory. So not all synapses are excitatory, meaning that they allow for sodium ions to come in. You have the action of inhibitory synapses, which take the membrane potential away from that action potential threshold. Now, these inhibitory synapses exert powerful control over neuron output, and these are definitely important because you need a balance of excitation and inhibition. If you had nothing but excitatory synapses, your brain would be in a constant state of seizing. Too much electrical activity. So, startle diseases are a class of diseases which show genetic mutations affecting the receptor for glycine. This is an amino acid neurotransmitter. Humans and in mutant mouse spasmodic, one amino acid is coded wrong, and a chloride channel opens less frequently when exposed to glycine. So glycine is normally going to be inhibitory, allowing for chloride to open, and for chloride channels to open, and for chloride to flow in. However, with this one amino acid coded wrong, your chloride channel opens less frequently, so you have less inhibition. So this mutant mouse, which is known as spastic, and in a strain of cattle, lower number of glycine receptors are actually seen to be expressed. So there is a delicate balance that's needed between excitation and inhibition in the central nervous system. And strychnine is an antagonist of glycine at its receptor. And one thing that I like to mention is, this is a spoiler alert from Game of Thrones, but strychnine poisoning could be a reason why King Joffrey died. So sorry if I spoiled that for you, but this is what's known as the Strangler. And there's a really cool video, and I'll try to see if I can find a link to it and I can share it with you all. But looking at the effects of strychnine poisoning in the central nervous system. Now, excitatory versus inhibitory synapses bind different neurotransmitters and allow different ions to pass through these channels. Inhibitory bind GABA or glycine and will allow for chloride ions to flow through. Excitatory can bind numerous different neurotransmitters such as acetylcholine and glutamate and will allow for sodium ions to flow through. Now, when the membrane potential is less negative than resting state, remember that we said that resting state is negative 65 or negative 70, give or take. This is a hyperpolarizing IPSP. So an inhibitory postsynaptic potential is an example of a hyperpolarization. Now, what's known as shunting inhibition is when the synapse will inhibit current flow from the soma to the axon hillock. So if we have an inward movement of negatively charged chloride ions, this is formally equivalent to an outward positive current flow. It's sort of like cutting a big hole in a leaky garden hose, where you're going to have this current flow take the path of least resistance. So IPSPs actually have the ability to reduce the size of excitatory postsynaptic potentials, and this makes the postsynaptic neuron even less likely to fire an action potential. So here we have an excitatory synapse that's active right here. We record the membrane potential. We see an EPSP occur. Now again, we have a decrease in the strength of the signal as we travel down the axon, or as we travel down the dendrite, sorry. Now, in the case of shunting inhibition, if we have an inhibitory synapse active right here, this will actually lead to the membrane potential even being less. So here we have the EPSP that travels, and it still has some form of strength when we measure the membrane potential in the soma. However, if we have an active inhibitory synapse which allows for chloride ions to flow in, you see that this membrane potential from this original excitatory synapse is essentially gone. So excitatory synapses, examples of glutamate, are Gray's type 1 morphology. Remember that this is asymmetrical membrane differentiations. And inhibitory synapses will display Gray's type 2 morphology, and these are GABA or glycinergic synapses. So in addition to dendrites, these will tend to be clustered on the soma and near axon hillock, which is a powerful position to essentially influence the activity of that postsynaptic neuron. Now, one important idea in synaptic transmission is the idea of neuromodulation, and we see this beautifully examined in G-protein coupled receptors, or GPCRs. So synaptic transmission that modifies the effectiveness of EPSPs generated by other synapses with transmitter-gated ion channels is an example of modulation. So a great example is activating the norepinephrine beta receptor. Now the norepinephrine beta receptor is going to be a receptor that will bind a molecule of norepinephrine. Now what happens when norepinephrine binds to this receptor activates its G protein. So we saw that that is what occurs in G protein coupled receptors, the activation of a G protein. Now what happens when this G protein is activated is it activates an enzyme. This enzyme is known as adenylyl cyclase. This will lead to the conversion of ATP into cyclic AMP, which is a second messenger. Now, the cyclic AMP is free to diffuse within the cytosol. It's free to go about every which way it wants in the cytosol. Now, cyclic AMP can stimulate another enzyme known as protein kinase. This protein kinase, and kinase is an example of a protein that is able to phosphorylate different channels or proteins. This can lead to a phosphorylation on other cell proteins, which can then change those proteins' conformations, thereby changing its activity. So it's able to, just one binding of a neurotransmitter onto one receptor is able to modulate the activity of channels and chemicals that are away from its site of activation. So chemical synaptic transmission displays a rich diversity that allows for several and a large number of complex behaviors. So chemical synapses provide explanations for drug effects, since drugs exert their effects at those chemical synapses, whether those drugs are going to stimulate receptors or whether they inhibit receptors. You're going to definitely have some form of effect on behavior and mental status. Defective neurotransmission is the basis for many neurological and psychiatric disorders where you have either an overexcitation, too much inhibition, too much release of neurotransmitter, not enough release of neurotransmitter, etc., that can play a key functional role in different mental illnesses. And understanding chemical transmission is ultimately the key to understanding the neural basis of learning and memory. And as you go on throughout this course, even looking at the sensory systems, it's going to be very important to be able to understand the role of chemical neurotransmission. And even when you go into Psych 376, you're going to be applying a lot of the knowledge that you learn in this class to neurotransmitter systems that are going to be impacting psychiatric conditions and other behaviors, including learning and memory, hunger, sleep, etc. So in the next chapter, what we're going to do is we're going to look at all the neurotransmitter systems in a lot more detail, talk about their synthesis, their breakdown, their transport, etc., so that you have more of a grand picture about how all these intricate parts are coming together to produce an experience. So I'll see you in the next video.